My name is Brad Howard. I'm a graduate student in Dr. Chris Lawrence's lab. I guess this is the Lawrence block today. I'll be presenting a poster this afternoon called The Holy Grail of Fungal Innate Immunity, more specifically chitin recognition and signaling in human airway epithelial cells. So broadly, I'm interested in human innate immunity and how fungal allergens interface at the host pathogen interface, and particularly at the mucosal surfaces in the lung. Over the past year, I've been studying a molecule called chitin, which is a really ubiquitous homopolymer of glucosamine, which is also an integral component of the fungal cell wall. So, there we go. So we know that chitin fragments initiate immune responses in the lung and induce inflammation. One of the big driving questions in fungal innate immunology at the moment is how is this actually carried out? So what is the receptor in humans and in animals for chitin and the downstream signaling processes that happen once that receptor is triggered? So leveraging what we know in plants, we've actually, in the human genome, found 4 genes whose protein products have predicted chitin binding properties. And 2 of these also have predicted transmembrane domains. So these were attractive targets for us in looking for a protein that may be a receptor for chitin in humans. So over the past 6 months, I've been doing some RNA interference studies. And it appears as though one of these transmembrane proteins is critical for not only sensing chitin fragments, but more importantly, it's also critical for sensing at least 2 pathogenic fungi that we've tested. So it's critical for sensing and initiating an inflammatory cytokine response. So also at this point, data I'll present today, we have some preliminary but very promising data that shows this protein does bind really well through isothermal titration calorimetry to chitin fragments in vitro. So we're moving forward on that as well. So yeah, come see my poster this afternoon. Thanks.